Welcome to the Live to 110 podcast. My name is Wendy Myers, and you can find me at liveto110.com. And you can find this video podcast on the YouTube channel, Wendy Live to 110. Please go there and subscribe. And you can find the video also on the corresponding blog post on my website. Today we have Michael McEvoy on the podcast. He's going to be talking about methylation and genetics and how they're interrelated. A lot of people are confused about methylation, and it's something that I think everyone looking to improve their health needs to learn their status. Are you an over-methylator or an under-methylator? It can have profound impacts on your health and any diagnosis that you may have. We'll also talk about genetics, the 23andMe test, and any kind of health apps. Michael has his own app that you can run it through to learn about your genetic SNPs and what consequences those SNPs have on your health. So lots of great info on the show today. I am so excited for the launch of my Body Bio Rehab program. You can learn more at bodybiorehab.com. And I wanted to create an online program like this that address the very basics of health, the basic things that you need to do to live a long, healthy life. And this program contains 5 modules. There is one on diet, the type of diet you should be eating, exercise, what type that you should do and how much, stress relief. I think stress is one of the #1 killers, and you need to have lots of things in your toolkit to reduce stress. Then we have sleep. Many people are not getting adequate sleep, and you can't heal your body and you can't detox and de-stress, etc. if you're not sleeping properly. So I talk about the importance of that and lots of simple tricks that you can do to improve sleep, what types of supplements improve sleep, which ones are the best. And then I talk about detoxification, the most important pillar. When people are daily exposed to thousands of toxic chemicals, it's estimated that there are 80,000 chemicals in our environment. And when we are exposed to all kinds of heavy metals, mercury, cadmium, lead, etc., these need to be detoxed from the body or they will eventually cause disease. And many practitioners, many health practitioners and doctors are not paying any attention whatsoever to detoxification. It continues to blow my mind because in the coming decades, this is going to be more and more important, and we will see an increase in diseases, the increase in prevalence of diseases because of heavy metals and chemicals. So you have to learn how to detox and have a lifelong detoxification plan. And so that's what I teach you in Body Bio Rehab, how to completely make over your body chemistry from the inside out. So learn more at bodybiorehab.com, and it's only $49. I wanted to make it very affordable for many people who maybe can't afford to work with me or people just want to enhance their experience working with me. It's an amazing program that I've spent about 6 months developing, so enjoy it. Our guest today on the podcast is Michael McEvoy. He is a functional diagnostic nutritionist, an FDN, through the Functional Diagnostic Nutrition Institute. He is certified in hair mineral analysis per Dr. Lawrence Wilson's protocols, like myself. Michael is a certified nutritional consultant and a certified metabolic typing advisor. Michael has studied advanced functional blood chemistry analysis, cell membrane dynamics, lipids, and the profound body of work of Emmanuel Ravici, MD. Michael is on the staff of the Functional Diagnostic Nutrition Institute, specializing in research and development. He is currently engaged in a 2-year course in professional herbal studies through Michael Tierra's East-West School of Planetary Herbology. Michael is a dedicated health nutrition professional who offers a wide spectrum of services for national and international clientele. Michael, thank you so much for coming on the show.
Thanks, Wendy, for having me. Great to be here.
So we're going to talk about methylation and genetics, but can you tell the listeners first a little bit about yourself and how you got into methylation and genetics, et cetera, and your website?
Well, I've been a health practitioner for the past decade, and I have developed a entirely web-based nutritional consulting practice. I am the CEO and founder of Metabolic Healing Inc., and our website is www.metabolichealing.com. And we work with a variety of different types of clients. We have clients that are very sick with chronic illness, chronic fatigue, adrenal and endocrine problems, methylation dysfunction, et cetera, et cetera, et cetera. And so we've found that there's definitely an integrated approach that can be used when helping to deal with a lot of these types of issues. And we've been doing this for several years now, and we've focused a lot of our attention in the last few years specifically on individuals who have problems with methylation in particular. And oftentimes these kinds of problems manifest with certain symptoms such as chronic fatigue, autoimmune problems such as Hashimoto's thyroiditis, other types of chronic gastrointestinal inflammatory problems, cardiovascular disease, especially if there's a familial risk for cardiovascular disease. So a lot of these issues overlap, not surprisingly, once you start realizing that the methylation cycle is directly related to each of these types of problems.
Yeah, being a health practitioner myself, I decided that I had to add genetics and methylation to my practice because it dramatically affects people's health outcomes and their ability to detox, and so many factors can be affected by this. And you can't really help people unless you're attending to these health issues. So let's talk a little bit about what exactly is methylation for anyone that doesn't know what that is.
So methylation it's actually a biochemical pathway that's happening in every cell of the body, something like 1 billion times per second in every cell of the body. You could think that that's quite a dance of activity. And the methylation cycle, it is so interconnected with so many things that the body does, from heart health, cardiovascular health, to endocrine function, to neurotransmitters, so affecting mood and behavior. A lot of people with anxiety and depression and bipolar disorder have problems methylating. So methylation is tied to so many different things, and as I said, it's a biochemical pathway that's going on in the body continuously. And we'll talk about genetics and the possible interaction that certain genes have on the methylation cycle. But methylation has gotten a lot of attention over recent years because there's so much scientific studies and literature that has been done on methylation, particularly regarding all of these types of health problems that we're seeing in the 21st century here.
Can you talk a little bit about overmethylation and undermethylation, and if you think it's really that simple?
Yeah, that's a good question. And so there's kind of this discussion of, well, what does it mean to be overmethylated? What does it mean to be undermethylated? Is there overlap? What exactly does this mean? So the methylation status is essentially, some clinicians like to classify these things as undermethylated, overmethylated. And so what does this exactly mean? So what is methyl? It's basically this chemical compound that's in the body. It's a very simple chemical compound, and it's basically it's found all over the body. A lot of different nutrients contain methyl. A lot of different important vitamins and minerals contain methyl. There's certain words called methyl donors, and certain nutrients, they'll donate a methyl group to another molecule, and it's kind of like this. It's kind of like a race, if you will, of nutrients. You can kind of imagine like you're handing off the torch to another runner in a race. In many ways, that's how the methylation cycle works, is where these nutrients are basically kind of dumping off their methyl groups to other molecules. And so that in this process, you've got all these things that go on, all these things that happen in the body. So some clinicians talk about undermethylated and overmethylated. Basically, these terms mean, in a nutshell, if you're undermethylated, it typically means that you have too few methyl groups. You need more methyl in your body. And people that are undermethylated, they tend to have and exhibit certain types of symptoms. They tend to be those that, for the most part, that suffer from depression. They may also have high levels of histamine. They may have environmental seasonal allergies. And a lot of methyl donors actually can help with those kinds of symptoms specifically. Overmethylators, essentially, they typically have too many methyl groups, and there's an overload of these methyl groups. There's too many of them. The body can't break them down, get rid of them quickly enough. And so typically overmethylators, they tend to exhibit anxiolytic symptoms, so more symptoms of panic and anxiety disorders. They may be estrogen intolerant. They may be very creative and artistic in nature. And so there's a lot of very interesting traits that are different among the over and under methylators. And it really comes down to how certain nutrients are being used and how enzyme reactions in the body are affecting these particular nutrients.
Once you know your methylation status, what do you do? Are there supplements that you should take, or foods that you should avoid, etc.?
Well, there can be, and it's certainly everything really depends on each person. There really aren't kind of broad one size fits all approaches. But once you know if you're undermethylated, what we do in our practice is essentially we do some specific functional lab testing that helps us to kind of see what a person's methylation status is. But we want to always kind of correlate and corroborate these lab tests with a person's clinical history, with their symptomology, just knowing more about the person in general. And so once we kind of get this good idea of what type of client that we're working with, then we can kind of say, well, these types of nutrients are likely going to be more useful for you specifically. So we try to get as individual and as specific as possible. So, for example, undermethylators tend to respond very favorably to zinc and B6 and methyl donors like betaine hydrochloride. And so often, and if somebody, other methyl donors like SAMe is another big one that especially works for the undermethylated depressed individuals. They have depression issues. So, but conversely, if somebody's overmethylated, they may tend to do much better with niacin or niacinamide, vitamin B3, folates, different forms of B12, because the different nutrients, they stimulate different enzymes in the body. And so if you're under over methylated, it really can come down to the kind of looking at these nutrient groups as ways to really kind of figure out how to best individualize it. So, so the answer is yeah, there can definitely be a lot of leeway to play with in terms of what individualized nutrient therapies we're looking at.
Yeah, I agree. It has to be highly individualized. There's so many variances between, within over and under methylation. So, what are the best tests to determine methylation status? How does someone figure out what they are?
So, first of all, I want to just say that in terms of methylation, there's kind of a lot of kind of overview about different tests that are out there, and the methylation status is probably the best test to kind of gauge if a person is over or under methylated is the SAMe to SAW ratio. And what does that mean? It's basically the S-adenosyl methionine to S-adenosyl homocysteine. This is the major methyl donor is SAMe, S-adenosyl methionine, and it basically creates this other nutrient called S-adenosyl homocysteine. Basically, the ratio between the SAMe and the SAW tells you a lot about the overall methylation status that the person has. There's other indirect ways of kind of assessing overall methylation function, and the whole blood histamine is one that we've been experimenting with and using in our own practice for quite a while now. And that's essentially because histamine is a very important, very interesting compound that gets produced in the body. Histamine, for example, it's an inflammatory compound produced by certain immune cells. A lot of people listening to this podcast may already know about low histamine diets and the problem with histamine-containing foods like red wine and aged cheeses and bananas and avocados. Some people think they may have histamine intolerance, and that very well may be true for certain people. But histamine is also a neurotransmitter, and by being a neurotransmitter, histamine affects the brain. It affects mood and behavior and mental health. It interacts with other neurotransmitters. Histamine also is important for gastric acid synthesis, so it's really hugely critical for gut function. And stomach acid is a major component of gut function. Histamine regulates the pituitary and the hypothalamus, and specifically the hormones that those glands produce. So histamine is actually being metabolized and degraded by methyl groups in the body. And so when a person has high histamine, the reason why they tend to be undermethylated is because the enzymes that metabolize and break these histamines down tend to be reduced. There's not enough of these enzymes present to break down the histamine. And so by kind of getting them on the protocol, it helps them to kind of modulate their histamine response more. So I don't know if I answered your question. I kind of went off on a little bit of a
No,
tangent
we
there.
like tangents. We love tangents.
Yeah, so histamine, it's an indirect biomarker of the methylation status, and we find that there's a lot of correlation between histamine, their methylation status, as well as what types of individualized nutrient therapies they tend to do well with. So those are the two major ways, but we always, in addition to looking at that stuff, we always want to look at some other evidence and some other lab tests. So in our practice, we utilize functional blood chemistry analysis, basically with every client that we work with. And this is a very different way of analyzing a blood test than from a kind of a Western pathological allopathic way of analyzing blood chemistry. We're looking to assess functionality. And so there's certain key nutrient deficiencies that often show up over and over again that you can screen for using routine blood chemistry. So we look at the alkaline phosphatase, the ALT, AST, GGT enzymes, liver enzymes. We look at the mean corpuscular volume as an indirect marker of B12 and folate utilization. We can look at serum zinc and copper is an indicator of zinc and copper metabolism, which that particular nutrient group, zinc and copper, can have a huge role in how the body functions, for better or for worse. Kind of one of the repeat offenders of nutrient imbalances in the body is low zinc, high copper. And that's especially true in certain client populations. People that have mood, behavior, and mental health issues, they tend to be copper toxic. And this is frequently found among people that have schizophrenia, bipolar disorder, mania, psychosis. High copper can also be a problem for as a potential cancer risk factor, because cancer thrives when there's too much copper in the body. Angiogenesis is dependent on copper utilization. So there's a lot, and PMS in women, that's another thing that tends to exhibit high copper, low zinc. So these nutrients directly have an effect on how we methylate and how our biochemistry functions. So we always want to be as thorough as we can. We want to look at specific lab tests that kind of give us clues about what a client nutritional protocol should look like.
Yeah, I agree with you. Copper is a huge, huge problem. I test, do hair mineral analysis and other blood work to determine copper dysregulation of the body, and it presents a whole host of issues for people, and including contributing to methylation issues. But let's talk a little bit about methylation and genetics and how they're related. Can you talk a little bit about that?
Yeah, so genetic testing has kind of opened up the floodgates in the last 5 years. It's kind of been this craze of genetic testing, probably due to the company 23andMe having kind of this direct-to-consumer genetic testing, which you can now screen for nearly 1 million genetic S&P singular nucleotide polymorphisms. And so as a result of that, there's been a huge amount of kind of attention that people have wanting to start looking at their genetic profiles and try to analyze where their heritable strengths and weaknesses are. So with any kind of a craze, that any kind of a health craze, I've kind of learned over the years that you have to really be kind of careful about it, because there's always going to be some truths, partial truths, half truths, and then just stuff that's just completely false. And I definitely feel strongly that this is true with regards to genetic testing. I do believe, and I think that there's no one that really can argue with this, that the heritable gene mutations can certainly cause problems and can predispose people towards adverse health effects and certain symptoms and diseases. I think that's totally true. But um, what I what my perspective differs is that genetic testing by itself, it really cannot be used to construct any kind of a meaningful therapy or protocol for somebody, primarily because your heritable gene status, it does not change, whereas your genetic expression can change. And that's not the same as saying that you're just, I have MTHFR C677T, I'm plus plus homozygous for that. That means I'm just going to start taking 5-methylfolate to try to quote unquote bypass that gene mutation. Well, some people do well with methylfolate, some people do horrible with methylfolate. And so the ready-made approach that so many clinicians use is, let's just try to supplement with this nutrient for this gene mutation. And it would certainly be wonderful if biochemistry was that simple, but obviously it's not. Things get a lot more complicated as you start really working in depth with somebody. You start to realize that there's a lot more things going on rather than just a single gene mutation. So we try to use, if we do find value in genetic testing, by all means, we do analyze certain genetic tests from a functional perspective, trying to understand where the weak points may be found, and then try to correlate those genetic test results with some type of other evidence, whether it be a biochemical test or clinical history, to try to get more of a holistic understanding of how to best use the gene test result. But there's a lot of clinicians out there that I feel are wrongly trying to use genetic testing as a sole approach to generate any kind of a protocol, and we just feel that that doesn't work. And we know that it doesn't work because we've tried it ourselves and we've seen the pitfalls of it. So um, genetics definitely can influence your methylation cycle. And I want to be clear about that not totally throw the whole genetic discussion in the garbage, because it's on the flip side of it, genetics can certainly have a tremendous influence on how we methylate. And there are certain genes that are operative in the body that directly influence how we methylate. And if they certainly can influence how our body functions. So just as kind of a very brief, how about 1 minute overview of genetics. So you basically have two different kinds of gene mutations. You have what are called somatic gene mutations, which basically are gene mutations that form throughout the course of your life. So if you're exposed to certain types of environmental chemicals or radiation, these can cause what are called somatic gene mutations. These somatic gene mutations are not passed down to your children per se. Heritable gene mutations are also referred to as germline mutations, and these are what are passed down from generation to generation. So these are two different kinds of gene mutations. So what does a heritable gene do? So for example, your genetics or your DNA is like a big biological template. It's a big instruction manual of how the body works. And we've got all kinds of different genes in our body. The instruction manual of our genes then gets basically translated into what's called RNA, ribonucleic acid. And the RNA is how we make these enzyme workers, these worker bees, if you will, that kind of direct the show of the biochemical activity that's going on all over the body. So there's all kinds of things that we don't even understand yet about genetics. You know, problems in the transcription process, the copying the DNA into the RNA. We don't understand all the different myriad of factors that can be influencing it all of the time. There's a big discussion about epigenetics and how epigenetics, which is regulating genetic expression, is possibly more important than just your gene status alone. And so, again, there's so many more factors to look at with regards to just saying here's a genetic test result, you have certain disease risk, etc. There's a lot more going on than that, and I think that our overall understanding of genetics is in its pre-infancy. That said, in terms of how genes affect our methylation status, they certainly can. And if you are, let's say, predisposed, if you do have the MTHFR gene mutation, if you're homozygous, you have a double mutation in that one of those key two alleles, then it certainly can impact how your body makes methylfolate. But at the same time, I want to give an example, and this has already been documented in the literature. Let's say you have a pair of identical twins. They have identical DNA. But twin number one consumes, is very healthy, takes extraordinary care of their health. Let's say they both have the MTHFR double mutation, but twin number one eats a whole food diet consisting of very high levels of natural folates found in foods. But in the flip side, twin number two consumes a junk food, processed food diet that's depleted in folate and other nutrients. I'm going to bet the house that twin number two winds up developing cardiovascular disease, whereas twin number one won't, because through the control of dietary and environmental factors, despite having identical DNA, twin number one has truly bypassed their gene mutation, whereas twin number two succumbs to their genetics. So this has already been shown that identical twins can have totally different outcomes with regards to their health, despite having identical DNA. And so we cannot just look at heritable gene status alone as knowing what's wrong with a person.
Yeah, I like looking at the genes to try to dictate how you should live your lifestyle and what kind of diet that you should potentially eat. Like for me, it was very profound when I learned I had a gene that reduces my metabolism of estrogen. So for me, that's a red flag. Maybe I need to reduce my caffeine because caffeine reduces the body's ability to metabolize excess estrogen. Really pay attention to my liver health and do coffee enemas, liver flushes, etc. because the liver metabolizes estrogen, etc. So I think it has really profound influence when people learn what their genetic susceptibilities are, and that can have a profound influence on their behavior and their lifestyle.
Yeah, it certainly can. But it's important to keep kind of a broader perspective in terms of really understanding what the body is doing at any given point in time. So if somebody does come to us with a certain gene mutation, we are always wanting to kind of put all of the evidence together about that particular gene. So if somebody has the MTHFR mutation, for example, there's about 20 questions that we need to ask to understand if that gene mutation is causing a major problem with that person or not. And we also have to look at certain lab tests that are going to help to validate whether or not that person is able to quote unquote bypass that mutation as they're currently living.
Yeah, yeah. Can you talk a little bit about people's methylation status and methylated B vitamins? For instance, many people that are over methylators have issues with certain methylated B vitamins. Can you talk a little bit about that?
Yeah, so there's definitely some B vitamins that contain methyl donors to them. So, for example, 5-methyl tetrahydrofolate is one of them. Another B vitamin is uh, methylcobalamin, which is the form of B12 in the cells. So some people actually have problems with these types of B vitamins because of the methyl. In the instance of methylfolate, it may not only be the methyl that people react adversely to, but it could also be the glutamic acid conjugates that are found in the folates in general. So if you look at the different kinds of folates that exist, you have folic acid, and then you have another one called folinic acid or calcium folinate, and then you have 5-methyl tetrahydrofolate. So these are all different kinds of folates. Folic acid, for example, is synthetic. It's not found in nature. It's actually poorly metabolized by the body. The liver doesn't contain enough of the FOLR2 enzyme to metabolize it in any active form, and so you wind up having an excess amount of folic acid that's circulating in the blood. Folinic acid is kind of a precursor to methylfolate, and it's typically tolerated well. Methyl tetrahydrofolate is the active form in the cells. It contains its own methyl group, but it also has is 9 or 10 glutamic acid conjugates. A lot of the common symptoms of methylfolate supplements is anxiety or mania even. And we've had people before taking like 15 grams of Deplin. They would come to us, which is the drug form of methylfolate, and they were hospitalized because of some psychotic episode after taking it. And you have to realize that methylfolate is loaded with these glutamic acid conjugates, which can increase the free pool of glutamate in the system, which is an excitatory neurotransmitter that can lead to a lot of those kinds of symptoms. So it does contain methyl as well, and sometimes people react adversely to methyl. Same thing with methylcobalamin, and sometimes you need to give another form of cobalamin like hydroxycobalamin, which is the form that's actually found in foods, or adenosylcobalamin, or even cyanocobalamin, which has kind of been demonized, even though it's an acceptable form of B12. So it really can, methyl can adversely affect certain people. It's true.
And
And
what
for
are,
other people, it could actually be very important for them, I should say.
yeah, because there's some people, but just about to say that there's some people that can't convert certain B vitamins in their raw form to the methylated forms in their body, so they need the methylated forms.
Another key nutrient that tends to not be absorbed well, or that's kind of one of these repeat offenders of nutrient deficiencies, is pyridoxal 5-phosphate. And so, just as a kind of a recap, you have nutrients, and then you have the coenzymated nutrients. And so, coenzymated is the actual form that the body uses for enzymatic reactions. So, in the instance of vitamin B6, the active coenzymated form is called P5P, or pyridoxal 5-phosphate. And from our own research, this is probably one of the most common nutrient deficiencies. And there are certain gene mutations that can predispose or prevent somebody from being able to convert pyridoxine into its active coenzymated form. And we just see over and over and over again how so many people do fantastic when they start supplementing with a little bit of P5P into their body.
Yeah, yeah, I've started taking that myself, and I've really felt a big difference in that. Also, a lot of people need B5 as well, especially if they have adrenal fatigue. They need to take extra B5 every day.
Yeah, pantothenic acid, B5, is definitely one of those kind of really hard to get nutrients, not because it's not found in foods, but because pantothenic acid as a molecule is very large, it's difficult to take up in high enough quantities in the intestinal mucosa. And so supplementing with B5 could definitely ensure that you're getting more of it into the body.
Can you talk about some of the tests that you can do to learn more about your genetics? We know about the 23andMe, and I have a lot of clients come to me and their doctor tested them just for MTHFR, which is probably not enough. Can you talk a
Right.
little bit more about that?
Yeah, so this is a great example is that you've got certain doctors and labs like LabCorp Quest that are basically running these relatively expensive gene tests, but they're only testing single nucleotide polymorphisms. So somebody would say, oh, their doctor quote unquote diagnosed them with the MTHFR C677T double mutation, but they didn't test any other SNPs that are related to that particular gene. So it's kind of like, well, what's the point? I mean, and this is just an illustration of how reductionistic, you could possibly get. Let's look at the smallest possible fragment on a chromosome and try to have any sense of idea about what to do based upon that alone. And so to me, that's a very fragmented, kind of reductionistic way of looking at genetics. A more holistic way of looking at the genetics just by itself is by looking at a bunch of gene SNPs that are related to one another, or that interact with one another, or are found on the same chromosome. So in the instance of MTHFR, I'm going to want to look at all of the other enzyme, the status of the other genes in that methylation cycle. So MTRR, or MTR, or BHMT, or MAT, or CBS. All of these other enzymes that are related. Nitric oxide synthase, that's another big one. Because methylfolate influences how we make nitric oxide. And so there could be all kinds of implications for what's going on if you have compound homozygous status. So let's say somebody is MTHFR++, but they're also NOS++. That means that not only do they may have a problem making methylfolate, but they also may have a problem making nitric oxide, for two reasons, because both of those genes are involved in nitric oxide production. So you got to look at kind of the whole kind of picture in terms of genetics. So in our practice, we have developed a reporting system, a nutrigenomics kind of analyzer reporting system that basically takes the 23andMe raw data file of our clients, and it automatically analyzes it and converts it into a PDF document that looks at about 200 of those singular nucleotide polymorphisms. And to me, that's a better way of kind of getting an overall profile of somebody's genetics if they want. And there's all sorts of apps that are out there that are doing these kinds of things as well. So, but we do recommend using 23andMe if somebody wants to test their genetics or wants to learn more about their heritable gene status. We recommend it because it's only $99. I
Yeah,
mean,
it's so inexpensive.
so inexpensive, and then it just costs another to run it through our app. It only costs another $37. So for $137, $136, you have a lot of genetic data on what your herited strengths and weaknesses are on a good list of some of the genes that you have. Obviously, not all of them because we don't have that yet. Nobody
Where?
does.
Yeah, no one does. Where can you find that app?
So we have a link to which I can provide you with, and you can give that to your viewers. It's basically very simple. You go, you pay the $37, and you get an automated email, and it just basically tells you how to upload your data from your 23andMe results. It's very simple, it's a 5-minute process, and that's it.
Okay, great. And that link will be in the YouTube description or the corresponding blog post on liveto110.com if you want it. I think that's incredibly valuable to do because you go to the 23andme.com and they only give you ancestral information like what your family history is, etc.
Right.
But they're not allowed to do health information anymore.
Yeah, exactly. And so even if people say, well, I went to 23andMe website and it says that they're not offering health data anymore, just ignore that. That's just their disclaimer. They're still testing for the whole genetic profile, the whole genome profile through their saliva testing. all you want is to get that raw data file. If you want to learn about your ancestry, that's all fine and great, and that's what they're basically offering. But if you want to really get kind of the meat of the report, you got to kind of dig a little bit. You got to kind of be a health detective and dig into that raw data file.
Are there any other genetic tests that you perhaps don't recommend?
That I don't recommend.
Yeah, because
Not
there's a lot
necessarily.
of genetic testing out there.
There's a lot. Well, there's a lot of different genes that are being analyzed for a lot of different things, and the question is, how, there, and then as we progress through the 21st century, there's going to be thousands more genes that are being studied, and so I'd say it's not things that I'd not necessarily recommend per se, but I'd say look for genetic testing that will kind of help to get more of a holistic perspective of how it really fits into your health, rather than the end all be all. And so I'd be wary of anything that claims to be the end all be all of anything,
Yeah,
because I don't think there is an end all be all to anything. Health is an endless unfolding process of discovery, and
yeah.
genetics is certainly one part of it. Can
Yeah, I think genes are a great way to look for healing opportunities, to see what further testing you need to do, or what more do you need to explore. It's just not a one size fits all. You have this gene that means this gene, take a supplement. It's just not quite that simple. Can you talk a little bit about can gene mutations be healed? Can you turn your genes on and off? I think it's something maybe a lot of people don't understand.
Well, heritable genes will never change. What you're born with and what you've inherited from both parents, that will remain forever in terms of the status of those particular genes. So, but the expression, if the quote unquote expression of certain genes, they can change, and that's kind of a really complicated discussion that really goes way beyond this call. But there is a way to kind of attenuate how our gene mutations are expressed. So, in the example that I gave before about the identical twins with identical DNA, yes, they have certain gene mutations which may predispose them towards a certain problem, but there's ways of kind of attenuating your genome through kind of looking at the epigenetic expression of how diet and environmental factors and stress really does play a huge role in how certain genes do get expressed. I should also point out that in terms of just somatic mutations, which are again those types of gene mutations that are not inherited from the parents but we have developed after conception, and this is basically how many forms of cancer are formed, is through somatic gene mutations. There's a lot of discussion about how environmental triggers are a huge component of this. So there's not a discussion, it's a known fact right now that there's a number of carcinogens that exist which have the ability to create various somatic gene mutations through the process of what's called mutagenesis. And so many people aren't even aware that common household chemicals are known mutagens. They're known to cause genetic mutations, so somatic genetic mutations. And so, for example, bromide, for example, is a mutagen. Mercury, cadmium are mutagens. The different types of chemicals that you find in household cleaning products can be mutagenic and carcinogenic. So this is really critical, and these things really need to be understood. And as Wendy pointed out earlier, bringing it kind of back to methylation, realize that our body's ability to repair our DNA, which to prevent continual gene mutations from spiraling out of control, somatic gene mutations from spiraling out of control, we have to methylate properly. And once we're methylating properly, one of the major things that we're doing is we're helping to regenerate and repair damaged DNA that happens on a daily basis for various reasons, for the aging process, etc. So methylation not only helps to regenerate our DNA, but it also is a pathway of what's called biotransformation. Helps to detoxify a lot of these carcinogens that we're being exposed to, that we're breathing in, that we're drinking, that we're consuming on a daily basis, even if we don't even know it.
Yeah, I think that's so important to talk about detoxification and how certain chemicals and heavy metals can affect our genetics. Because when our genes are copying, when they are transferring, the heavy metals and mineral deficiencies can interfere with how our genes are copied and cause mutations. It's not just that there's a defect in our body. Heavy metals and chemicals and mineral deficiencies are causing this. Can you talk a little bit about that?
Absolutely. And we need nutrients as the main fuel of every biochemical pathway inside of the human body, including the methylation cycles. Nutrients are not just things you take supplementing. People think, oh, I'm just going to take a supplement. and but take a multivitamin. Well, realize that those nutrients are actually the chemical fuel that your cells are using to work. So, and that's true of your food, obviously, is that's what the food and nutrients do. They fuel these biochemical processes that repair tissues, that repair damage to certain tissues and organs, so that we can kind of offset the effects of aging on our cells. So, if you open up any biochemistry textbook, for example, every single biochemical pathway that exists in the human body, every thousands and thousands of them, are all driven by nutrients, nutrient substrates, or nutrient cofactors or promoters. So all the B vitamins, all the minerals, all the trace minerals, macrominerals, these are all involved and operative in the biochemical processes, including methylation, as a way to keep the body operating. And certain gene mutations can influence how we use these nutrients, for sure.
Yeah, can you talk a little bit about what else you can learn from getting your genes tested? What are some of the things that people can find out by doing a 23andMe test?
So 23andMe is primarily using their genetic testing as a way to kind of learn about your ancestry, and this is kind of a really cool thing if you haven't done this before. It'll help to track where your family came from in certain parts of the world, certain parts of Europe, certain things that I learned about myself that I had no clue about before, about having Asian descent and having South American descent and sub-Saharan African descent. And where did it just kind of leads you into wondering, well, where along the genetic line did this all happen? So that's really one of the cool things that I think is really good about doing 23andMe, is it does give you that information.
Is there anything in regards to health that people can learn about? What kind of things they learn based on their genes?
Well, I don't know specifically if they're still doing any health related information anymore, because the FDA kind of put the kibosh on that. But they were for a while, and I don't know if anybody's already done it before the FDA gave them that notification. But there were certain quote unquote disease risk percentages that we're giving, and that's very controversial stuff. A lot of people think that I have such and such gene mutation, and therefore the only real solution to that is to excise my breasts and to remove organs to prevent diseases like cancer. That's a very controversial, really touchy subject, but I think it's one that's really important to kind of bring into the open, because as we learn more about genetics, we'll really understand that the heritable gene status is not the only thing that really matters, and that controlling gene expression through the epigenetic modification of our genome is just as, if not more important than the actual gene status itself.
Yeah.
And so I really think that our present understanding of genetics is still in its infancy. In 100 years from now, we're going to have a lot better understanding about what the genome really is and it's doing in the body. And even on a daily, monthly basis, there's so much research coming out on genetics and epigenetics that it's really mind-boggling.
So when someone runs their 23andMe through your genetic app, what kind of things can they learn? Could they learn about the GAD, where they have more anxiety, like some of the things like that?
So in our nutrigenomics profile, we screen for a couple hundred of these gene variants called S and Ps. And so here's the other thing, is that each gene has all these different variants of itself called S and Ps, or commonly abbreviated as S and Ps, which means singular nucleotide polymorphism. So, and then if you look at the literature, there are certain S and Ps that have been studied more than others as being really influential. So in terms of the MTHFR, for example, there's two of all the different S and Ps, the MTHFR S and Ps, there's about two S and Ps that have been studied, probably include more than 90% of the total research in that single gene are just those two S and Ps. So they definitely have a big wheel of influence. So that's true of other genes as well. So what you get with our report, you get kind of this overview of what the gene status is with certain S and Ps, and then you also get a description of what each S and P has been shown to do. So what we're really concerned with is the function, understanding the function of what each of these genes are operative, what they're doing in the body.
Yeah.
So for example, MTHFR, it's donating a methyl group for the purpose of remethylating homocysteine. And so we give this overview of what each of these genes does. You brought up GAD1, glutamate decarboxylase. This is the gene that converts glutamate into GABA, which is two important neurotransmitters that can go wrong if you have compounded mutations in these particular genes. So we talk about and also we're cite every gene that we're that the report assesses. It's just being kind of citations back to the scientific literature about what's known about that particular S&P.
Okay. Yeah, I learned with, I have a few of the GAD mutations, and that can cause people to have more anxiety, more addictions, and things, food or what have you, and that they probably need to take some GABA to try to regulate that since they're not really making enough GABA.
Or taking more thiamine and P5P, because those are
Yeah.
the cofactors for the glutamate GABA conversion.
Yeah.
And so if somebody were to come to me with a high frequency of GAD1 double mutations, what I'd want to do is I'd want to look at the key nutrients, such as so look at some lab testing that would tell us more about B6 deficiency or thiamine deficiency. So we might run a urinary organic acids test to get those indicators. And you could also look at like a neurogistics urinary neurotransmitter test to see what the glutamate and the GABA levels actually are. If you're seeing correlations there, then it's a good indication that there may be some problems going on with the GAD1 gene.
Yeah, it's so interesting that you can go down so many rabbit holes when you're looking at the genetics. I love it.
Yeah, it's really true.
So you offer several courses to health practitioners and to the public. Can you talk a little bit about those?
So over the past several years, since we've been working with individuals with various gene mutations and just as importantly, looking at certain functional lab tests that are associated with these gene mutations, or at least can be associated with them. Basically, we've over the last several years of working with this kind of in-depth client management, the culmination of which I've created a clinician master course, clinical master course called the MTHFR Methylation and Biochemistry Master Course, and it's essentially a 10-hour training program that is basically consists of two different modules. Module 1 teaches the clinician all about the major methylation-related genes. It also basically gives you an overview of what genes are for, how we convert DNA into RNA, and just kind of a very, very basic kind of genetic 101 assessment of all that. And then we look at an animated version of the methylation cycle and how all of these things are fitting together, so you can actually kind of see the dance of nutrients moving in an animated fashion. And then in module two is called Implementing Methylation in Biochemistry Specific Nutrition. And module 2 is all about the implementation of nutrient therapies that are specific for an individual. So this includes analyzing, not only using a gene test, but also looking at certain key functional lab tests that could be real deal makers with certain clients. And so we look at the urinary organic acids test. We study the functional blood chemistry analysis. We look at plasma amino acids. We look at histamine, zinc, copper, urinary pyrols. All of these other functional tests that from a clinical perspective can really be huge, very important for understanding what's going on with somebody. And so, again, it's kind of this really holistic interpretation of how to really best use a genetic test in combination with this overall holistic clinical investigative process. And so we go really in depth, and there's really no other course that exists that does this kind of material. The other thing I was kept in mind as I was creating this course was that I realized that all of the other methylation courses that exist out there are so genetically oriented. And I found that just in my practice alone, from a clinical perspective, trying to treat genetics, and I tried to do it that way for a long time, just try to use this gene mutation and use a genetic test to try to really get them on a certain supplement protocol without really paying any attention to these other important tests. And what I found was it doesn't work. It's just when the rubber meets the road, it doesn't work that way. So the best way to use a gene test is kind of in concert with all this other investigative process. And so this is truly a holistic course that will really empower the clinician to help their clients to adapt to their whatever their health lifestyle problems are, whatever their genetics are. This course will teach clinicians how to teach their clients how to adapt and how to get an ideal level of health despite their gene mutations.
You teach this course also at FDN, the Functional Diagnostic Nutrition. You teach a abbreviated version of that, correct?
Well, in the FDN module 2, so I'm basically on the staff as the head of research and development, or one of the R&D part of the R&D team with FDN, and I basically in module 2, FDN 2, I created a course which was specifically to teach clinicians about how to use the environmental pollutants test combined with the organic acids test, how to interpret the organic acids test. So that was kind of a very kind of beginning phase of that. But FDN practitioners, if there are any FDN practitioners that are listening to this, my methylation course is approved for CE units, for continuing education units, about
Okay,
10
great.
hours material.
Oh, fantastic. Yeah, it looks like an amazing course. Fantastic course. Yeah, because I was searching around for methylation courses and I came upon you, your website, and I thought it looked fantastic. And that's why I wanted to have you come on the show and talk about it.
Again, I'm not opposed to, I appreciate all of the other work that other clinicians are doing out there regarding methylation and MTHFR, but I feel that there's just kind of this overemphasis on genetics in general without necessarily looking at how to really do good clinical investigative work and use biochemical testing in concert with it. So that's what
I
you get in the course.
Yes, yes, yes. Yeah, well, I have a question I like to ask all of my guests. What do you think is the most pressing health issue in the world today?
Besides ignorance,
Yeah,
that
exactly.
may be the big one. I don't know. Well, that's a good question. And in the 21st century, I think that there's some unique things that we're really going to have to deal with that we weren't having to deal with even 50 years ago. I'd say that probably one of the most pressing concerns, in my opinion, is the prevalence of varying forms of environmental toxicity. And the reason I bring that up is that if you look at what humans were exposed to in terms of environmental pollutants 100 years ago, there was really no such thing as environmental chemicals 100 years ago. It was mercury and cadmium and lead and heavy metals, and we've known about the effect of toxic metals on health since antiquity for thousands of years. But the presence of environmental chemicals like bisphenol A, for example, glyphosate, these types of chemicals are very new. And the human body, I don't know if it can process or remove these chemicals to the degree that it needs to achieve an ideal level of health, or at least it makes it very challenging. And there's very little research that's been done on the compounded effect that environmental chemicals have. So we know that certain chemicals cause cancer, but we don't know what happens when you combine that chemical with 6 other chemicals or 10 other chemicals or 80 other chemicals, which in reality is what people are being exposed to today. We're not being exposed to 5 chemicals, we're being exposed to 80,000.
Yeah.
So to me, the pressing issue in the 21st century is how to adapt biologically, despite these myriad environmental toxins that are affecting us on a day-to-day basis. And I think that we're going to continue to see the rises of neurologically inflammatory diseases like autism disorder because of environmental toxins like mercury and other chemicals and heavy metals, but other types of conditions like Alzheimer's disease, cancer, Parkinson's disease, multiple sclerosis. I think these diseases are going to continue to be on the rise. And I think that environmental toxins, which are so ubiquitous in the environment, are definitely playing a huge role in gene expression and in the development of these types of problems.
Yeah, I completely agree with you, and that's why I advocate as much as I can the use of infrared saunas because this is one of the only ways that people can get these chemicals out of their bodies, sweat them out of the body because the liver can't handle them. The liver has never seen many of these chemicals, and our livers are so overloaded. You need to do an infrared sauna to bypass the liver and get these chemicals out of your body. I think it's the only hope, I think, for many people to live a long disease-free life.
Yeah, we frequently recommend sauna therapy to our clients, and there's been a lot of research going back more than 40 years now showing the actual benefits of sauna therapy. And for example, numerous toxic metals and chemicals have been shown to be increased in very high amounts via the sweat glands, even more so than the urine and through the kidneys and through the liver and gallbladder. So sauna therapy, just from the perspective of removing toxic chemicals and heavy metals from the body, certainly has its place. And in addition to that, sauna therapy has been shown to be very effective at raising nitric oxide levels, which is especially critical for anybody dealing with cardiovascular disease, diseases of neuropathy, problems with microvascular circulation, things like this.
Yeah, yeah, I think it's they're just the benefits are too numerous to list, even in 1 hour show.
Right.
But they're just they're incredible. I love my sauna. So, can you tell the listeners a little bit more about yourself and where they can find you?
So again, my name is Michael McEvoy, and I'm a nutritional consultant, and I'm the CEO and founder of Metabolic Healing. And we have an online web-based nutritional consulting practice and educational programs that we offer to clinicians as well. And our website is www.metabolichealing.com. That's metabolichealing.com.
Well, Michael, thank you so much for coming on the show. I really appreciate it.
Thanks, Wendy, for having me. It's great to be here.
And stay tuned, listeners, if you want to learn more about how to heal your health conditions naturally, detoxification, and the modern paleo diet, go to my website, live to110.com. Thank you so much for listening to the Live to 110 podcast.
I think that's where it starts. And I'm being cheeky, but yes, it's also true. This has evolved as very much a life path for me. My journey has lots of twists and turns along the way, and it never really made sense until I got into or began stepping into purpose. And stepping into purpose both from a corporate perspective, but more so from a personal perspective in terms of really, really owning how I see the world, how I view myself. I'm a little bit, anybody that knows me, I'm a little bit different in that I've always been, I guess, as a child was called overly sensitive. As an adult, I would say it's very introspective. I watch my own life as a TV show, and I really evaluate every choice, every mistake, every challenge, every success, to see what did I do to contribute to that? What could I have done differently? What did I learn from it? I have now the tools to watch from that perspective, as opposed to being hypercritical, overly, self-critical, when you're younger, maybe, challenging yourself or being overly sensitive, things like that. I think, as you grow older, and especially the path that I've taken to really learn, to go inward, to read, to build on top of a psychology background, which when I went down that path, I didn't realize I'd end up where I am now. So that kind of all comes together after a whole series of changes. I started out as a child performer, singer, vocalist, band geek. Saxophone for most of my childhood. I stopped that and I went out for sports. And for all the reasons that many go through, the bullying, the lack of understanding, the wanting to be part of a different crowd, and that led me, though, into a successful avenue, which sports took me all the way into college, and ended up in business from there. And so there's a whole series of very direct twists and turns that really embody where I'm operating from now, which is embodied in the two words that accompany our show and the business that I build from, which is life changes.
Yeah, tell us about that radio show. I was very, appreciative. You asked me to be a guest on your very popular radio show. Can you tell us a little bit about that and the genesis of beginning that show?
Thank you. Well, the show has evolved over the past 6 years now. I was gifted the introductions of two beautiful people in Filippo